Hi, my name is Siti. So today I'm going to presenting about nucleic acid as a drug target. Okay, what is the nucleic acid? Nucleic acid as a drug target refer to the strategy of designing therapeutic agents that directly interact with DNA or RNA to modify genes expression, block pathogenic sequence, or inhibit replication and translation of disease causing elements like viruses or cancer genes. Unlike traditional drugs that target proteins, these drugs aim to the genetic instructions themselves. Okay, as we can see here, got two type of the nucleic acid target, which is DNA and RNA. And let me explain about DNA first. Okay, what is the DNA? DNA means deoxyribonucleic acid. DNA targets specific sequence or structures like promoters and oncogenes. It is block transcription or induce DNA damage in cancer cells. Example, cisplatin bind to DNA, causing cross-link that kill rapidly dividing cells. The next is a ribonucleic acid, which is RNA. It is target to mRNA, microRNA, or viral RNA. Block translation alters placings or degrade pathogenic RNAs. Example, antisense oligonucleotides assess bind mRNA to prevent protein production. Structure human cells called nucleus. Nucleus contain genomes which is split to 23 pairs of chromosomes. Drugs which act directly on nucleic acid such as disrupt its replication, disrupt transcriptions, and disrupt translation. So now what's going on? Okay, nucleus contain the genome which is split between 23 pairs of the chromosome. The DNA are sections called genes. Genes making protein, and the genes switch on RNA polymerase attached to the genes. Messenger RNA move out of the nucleus into the cytoplasm. Protein factories ribosomes bind to the messenger RNA and will produce amino acid. Protein synthesis is the biological process by which cells build proteins which are essential for structure, function, and also regulation of the body's tissues and organs. It is involved decoding the genetic instructions stored in DNA to create specific proteins. Protein synthesis occurs in two main steps, which is transcription and also translations, to produce a chain made up of amino acid to proteins. It is important to human body to make enzymes, hormones, and antibodies, and also repairs and build muscles and tissue, essential for cell growth and function. So now we move to the next slide. Okay, for the next slide, I will explain about a chemical structure of nucleic acid. The chemical structure of nucleic acids, DNA and RNA, is crucial to understanding how they function as drug targets. Drugs that target nucleic acid interact with their chemical components such as nitrogenous bases, sugar phosphate backbones, or unique 3D structures. Okay, DNA, intercalating agents, alkylating agents, and chain cutters. rRNA, nucleotides, the building blocks. Each nucleotide is made of nitrogenous base, which is purines and pyrimidines. Okay, beta sugar, which is deoxyribones in DNA and ribose in RNA. Phosphate group links the 3 of carbon of the one sugar to the 5 of phosphate of the next nucleotide form the sugar phosphate backbones. Nucleotides of DNA 4 nucleoside deoxyadenosine, deoxydenosines, deoxyguanosine, deoxythydines, and deoxythymidines. Nucleoside building blocks are joined together. Phosphate group link the fifth hydroxyl group of the nucleoside unit to the 3 hydroxyl group of the next level. Okay, ribonucleic acid of RNA is similar to DNA with the following exceptions. Ribose is used instead of deoxyribose, and uracil is used rather than thymine. Okay, that's all from my presentation. Hope you enjoy my presentation. So, thank you. Have a nice day.